So we have next, we have from MD Anderson. It is really kind of a super treat for us to be able to get this amazing speaker. All right, Dr. Ferdinandos, Skoulidis, whose name I'm totally butchering, and I'm so sorry. I didn't write down a better role of it, and he's smiling at me, and I appreciate your kindness. He received his medical degree from Aristotle University in Greece in 2000, and his PhD. Well, that's an amazing place to be, and a PhD from the University of Cambridge in UK, and subsequently a C. Oh my goodness, I'm going to have to make better notes next time. Dr. Skoulidis' laboratory is focused on unraveling the molecular and clinical heterogeneity of KRAS mutant non-small cell lung cancer. And heterogeneity just means differences, y'all. Don't like get freaked out about it. And developing novel therapeutic strategies to target our common enemy, KRAS. So with a great privilege and such a pleasure to welcome you this morning. Thank you for joining us.
Morning, and thank you for having me. Thank you, Terry, and thank you, KRAS Kickers. Let me share my screen with you. Hold on. Share screen. All right. Can you all see it? Yes, you can. All right, so it's an absolute pleasure to be presenting to this group, to this very active group. It's doing a fantastic job. The title of my talk is going to be Alive and Kicking, Current and Emerging Strategies to Tackle KRAS Mutant and Non-Small Cell Lung Cancer. I am an associate professor here at MD Anderson. I'm a physician scientist, which means I spend part of my time treating patients with non-small cell lung cancer, many of them with KRAS mutant non-small cell lung cancer, but I also spend most of my time running a lab trying to understand how best we can treat this disease. So these are my disclosures. So as you know, KRAS mutations represent the most prevalent oncogenic driver event in non-squamous non-small cell lung cancer. In this pie chart, KRAS mutant non-small cell lung cancer always represents the biggest part of the pie, the biggest piece of the pie. Approximately 30% of all non-squamous non-small cell lung cancers will harbor a KRAS mutation. And within non-small cell lung cancer, KRAS mutant non-small cell lung cancer, by far the most common KRAS allele is KRASG12C. It's approximately 41% of the total, followed by G12V and G12D and G12E. So KRASG12C mutations overall are found in approximately 1 in 7 to 1 in 8 non-squamous non-small cell lung cancer. It's a very big part of the overall patient population. One important point that I would like to make from the beginning, and this will become even more relevant as the talk advances, is that KRAS tumors are very different from each other. They are, as Terry mentioned, heterogeneous. That means that one patient's KRAS mutant tumor behaves very differently than another patient's KRAS mutant tumor. And there are two major types of determinants, two major factors that determine why one patient's KRAS mutant tumor is different from another patient's KRAS mutant tumor. One of them is the different KRAS alleles. And this is, for example, KRASG12C, KRASG12V, KRASG12D. This is particularly relevant nowadays where we have allele-specific inhibitors that will only work for one specific KRAS mutant allele, but not for the other. But also, and perhaps even more importantly in trying to decide what makes KRAS mutant tumors different from one another, is mutations not in KRAS itself, but what we call co-mutations. So these are mutations in genes that happen together with KRAS. Dr. Dare just told you that KRAS itself is likely not enough to transform a cell to make it cancerous. It requires additional mutations. So it's these additional mutations that happen that to a large extent determine the heterogeneity of KRAS mutant and non-small cell lung cancer. And several years ago, when I first joined MD Anderson in 2015, that was the key question that I wanted to answer. What makes one patient's KRAS mutant tumor different than another? So we found that there are three major co-mutations, three major genes that can classify KRAS mutant tumors into three groups that we now call KP. These are tumors that have got mutations in KRAS, but also in a second gene, P53. KL, these are tumors that have got mutations in KRAS and in STK11 or LKB1, so a different tumor suppressor gene, and KC. These are tumors that have got mutations in KRAS and inactivation of the CDKN2A and CDKN2B tumor suppressor gene. And this is important because approximately, especially mutations in STK11, as well as a second gene that happens frequently with STK11 called KEAP1. This is important because approximately 1 in 3 KRAS mutant tumors will have a mutation in either STK11 or KEAP1. So how does this affect the biology of the disease and how does it affect response to treatment? What I'll say from the beginning is that it has a profound impact on the response of KRAS mutant non-small cell lung cancer to various standard of care therapies. So today, before the discovery of KRAS inhibitors, how would we treat a patient with a KRAS mutation with metastatic lung cancer? We would use immunotherapy, the combination of immunotherapy with chemotherapy, chemoimmunotherapy, or platinum doublet chemotherapy. And if they progressed on that, we would use second-line chemotherapy with docetaxel. Where here we are showing that clinical outcomes of KRAS mutant tumors, non-small cell lung cancer, with immunotherapy depend to a large extent on the co-mutational status. So tumors that have got the KL tumors, the ones that have got mutations in STK11, have got worse clinical outcomes with immunotherapy, whereas immunotherapy works very well for patients that have got a mutation in KRAS and a concurrent mutation in P53. So this is an important consideration as we assess the impact of different therapies, both standard of care therapies, but also the new KRAS inhibitors on clinical outcomes. So with that, I'm going to move to the KRAS inhibitors. And of course, this represents, and KRAS was considered undruggable for almost 40 years due to a variety of reasons, including the shape of the protein, as well as its affinity for GTP and GDP. This all changed. The watershed moment was in 2013 when Dr. Shokat's group identified compounds that were able to bind to covalently, so irreversibly, to the cysteine at position 12 in KRASG12C and occupy an induced pocket, a pocket that was previously unappreciated, a groove that was previously unappreciated. And by doing that, it locked KRAS into an inactive conformation, the GDP bound form of KRAS that then shuts KRAS signaling down and triggers proliferative arrest as well as the tumor cell death, apoptosis. And since then, we know that multiple rounds of medicinal chemistry have followed, and we now have clinical grade inhibitors such as Sotorasib, adagrasib, and various others that we're going to talk about. So with that in mind, these are the pivotal studies for the two drugs, for the two frontrunners. Sotorasib, of course, a drug that I had the privilege to participate in its clinical development. This drug got FDA approval for the treatment of patients with advanced KRAS-G12C mutated non-small cell lung cancer that have failed at least one prior line of systemic therapy, with an objective response rate of about 37%, median overall survival 12.5 months, median progression-free survival of 6.8 months. More recently, we saw data for adagrasib, another mutant selective inhibitor that shows very similar efficacy to Sotorasib. These were data published in the New England Journal of Medicine by Passiyani and co-authors in July of this year. And on the basis of these results, adagrasib was granted FDA breakthrough therapy designation in June 2021. More recently, in just a couple of days ago, we also had another major milestone in the treatment of KRAS-G12C mutant non-small cell lung cancer because we had the results of the first randomized phase 3 clinical trial of Sotorasib versus docetaxel. So this is not a single arm study. This is a randomized study where 345 patients are split 1-to-1 to either receive Sotorasib at the recommended phase 2 dose of 960 milligrams once a day, or docetaxel 75 milligrams per meter square, IV once every 3 weeks. And the conclusion from the study, this was a positive study. Sotorasib resulted in a 34% reduction in the risk of disease progression, the tumor growing, or death compared with docetaxel. So this was a positive study, and this is the first positive randomized phase 3 clinical trial for any KRAS G12C inhibitor. This is also a big moment. Responses to Sotorasib in CodeBreak 200, the phase 3 trial, were 28% compared to 13.2% with docetaxel. So it more than doubled the likelihood of achieving tumor shrinkage. And responses to Sotorasib occurred more rapidly. The median time to response was 1.5 months, 1.4 months, and were more durable. They lasted longer, and they were also deeper. In other words, the tumor shrinkage was on average higher with Sotorasib than it was with docetaxel. Let's look a little bit about the safety of these drugs, which I know is something that quite rightly concerns patients significantly. So both Sotorasib and adagrasib are by and large safe drugs. Sotorasib, this is the safety of Sotorasib. The majority of treatment-related adverse events were low grade, and they were managed with standard supportive measures or dose modification, interruption, or dose reduction. Only 7% discontinuation of drug due to treatment-related adverse events. For adagrasib, a slightly higher rate of GI adverse effects, but again, the rate of discontinuation of the drug approximately 7%. Another important thing that is the intracranial activity or the potential intracranial activity of these drugs. And these are retrospective data from CodeBreak 100. These were patients that had previously treated brain metastasis, and we were looking at whether Sotorasib was able to maintain those responses or induce radiological responses. But these were patients previously treated. And in this retrospective analysis, approximately 13% with evaluable brain metastasis had evidence of an intracranial response. More recently, we saw data for adagrasib, both in previously treated brain metastases, where approximately 1 in 3 had an intracranial objective response, but also more importantly, and this is the lower part of the curve, in patients whose brain metastases were not previously treated. So in this study, among patients with previously untreated brain metastases, adagrasib yielded 32% intracranial response with a median intracranial progression-free survival of 4.2 months, which is certainly encouraging. I'm going to spend some time talking a little bit about other KRAS inhibitors, which are entering or advancing in clinical development. So this is JDQ443, developed by Novartis. So we saw results earlier this year at the American Association of Cancer Research meeting in spring from the CONTRAST-01 clinical trial of JDQ443 monotherapy. And again, some encouraging news there. The safety profile of the drug appears pretty good. And the efficacy of the drug also looks highly encouraging with an objective response rate of 45%. This includes both confirmed and unconfirmed responses, and perhaps an even higher objective response rate among patients with KRAS-G12C mutated non-small cell lung cancer treated at the recommended phase 2 dose of 200 milligrams twice a day. Obviously, we need to see more data and more advanced data, but this is certainly promising. And we also saw at this year's World Conference on Lung Cancer in the summer in Vienna, data from GDC 6036. This is from Genentech Roche. And again, the conclusion is similar, that this is the safety profile of the drug certainly is compatible with what has been reported for the other KRAS-G12C inhibitors. And again, the efficacy is very promising with an unconfirmed objective response rate of 53%, confirmed objective response rate of 46%, and what appears to be durable responses, although of course the follow-up is relatively limited. And also another KRAS G12C inhibitor that was, this was presented again at the World Conference on Lung Cancer by Dr. Xun Liu. So this is the safety and efficacy of D1553, a phase 1 clinical trial. Again, the safety data is reasonable, perhaps a little more ALT/AST elevation, but the objective response rate, 37%, disease control rate, 90.3%, median duration of response, 5.4 months. We need more data. This is a relatively immature cohort, so we'll see whether this evolves as time passes, but this certainly is in keeping with the clinical efficacy of other inhibitors. One important point I mentioned at the beginning, that co-occurring alterations, co-mutations, impact clinical responses to standard of care therapies. So this is important because they may also impact clinical responses to G12C inhibitors. So this is early data from both the CodeBreak 100 study as well as the Crystal-1 study for adagrasib, where you can see that perhaps the objective that the likelihood of tumor shrinkage with G12C inhibitors to some extent depends on co-mutations. You can see, for example, that there's a lower objective response rate in patients with KEAP1 co-mutations, which is one of the key co-mutations that occurs with KRAS, perhaps suggesting that we need different approaches or more enhanced approaches, combination approaches in this genetic subset. So with that in mind, I am moving to the next chapter, which is we now know that KRAS inhibitors, which were not previously possible, are a reality. KRAS is druggable, and the objective response rate is in the region of perhaps 1 in 3 patients, 30 to 40% across different clinical trials. This is very encouraging. This is great, but of course, it's lower than other targeted therapies. So we are kind of thinking that combinations of G12C inhibitors with other drugs might represent the optimal therapeutic approach for the future to enhance both the likelihood of achieving a response, but also the durability of response. And you can see here in this great review from Cancer Discovery, both upstream components as well as downstream components of the KRAS signaling pathway, and suggesting different potential points of rational intervention with additional drugs in order to increase the efficacy of mutant selective inhibitors. And so we can either target upstream components of the pathway, such as the SHP2 phosphatase or the SOS1 guanine nucleotide exchange factor. These factors affect the balance between GDP-bound KRAS and GTP-bound KRAS, therefore potentially affecting the amount of drug, the amount of KRAS that is available to bind to mutant selective inhibitors such as Sotorasib or adagrasib, but also downstream components of the pathway, both in the MAP kinase pathway, for example, RAF, MEK, or ERK, as well as in the PI3K AKT pathway. And on the basis of this conceptual framework, all the different KRAS G12C inhibitors are currently being combined with different agents targeting different nodes in this pathway, such as EGFR inhibitors, SHP2 inhibitors, SOS1 inhibitors, as well as more conventional treatments such as chemotherapy. I'm going to present very briefly some data on most recent data with some of the most promising combinations that this is the first data that is emerging. So this is data presented at World Lung for Sotorasib and the SHP2 inhibitor RMC4630. So the combination, and this is from CodeBreak 101 and subprotocol C. The early readout from this trial is that the combination is tolerable. There are some side effects that one needs to be aware of. About approximately 30% of patients have got edema, fluid retention, but by and large, the combination is tolerable. And in patients that had not previously been treated with a KRAS G12C inhibitor, KRAS G12C inhibitor naive, the early efficacy signal is positive. The objective response rate was 50% among KRAS G12C inhibitor naive patients. And in the 2 highest doses of the SHP2 inhibitor that were studied, 3 out of 4 patients achieved an objective response. So this is, of course, early data, limited number of patients, but nonetheless positive data. And a study is underway to further define the efficacy and safety of this combination in patients with metastatic KRAS G12C mutated non-small cell lung cancer that were treatment naive. Other combinations had been tried before. So this is the combination with Sotorasib plus afatinib, the pan-HER inhibitor. So this what was completed previously. This was an active combination, but it also led to significant overlapping toxicity, mainly GI toxicity, potentially limiting the efficacy of the combination. So this is something that we always need to take into consideration when we try to combine KRAS inhibitors with other drugs. What are the overlapping toxicities and what is the optimal window what's the optimal therapeutic window? And a similar conclusion from when Sotorasib was combined with trametinib. It was doable, but the objective response rate of this combination was a little bit lower, 20%. One important point, KRAS G12C inhibitors are being combined, and this is one of the most exciting and one interesting areas for future work with immune checkpoint inhibitors. Why is this important? This is important for a number of reasons, because at least in mouse models, the efficacy, the ability of a KRAS G12C inhibitor to induce a long-lasting remission, in other words, not just initial shrinkage, but really a cure, if you want, in the mouse, is only achievable in mice that have got an intact immune response. If the same experiment is performed in mice that are immune deficient, that do not have an intact immune response, you see an initial shrinkage of the tumor with many of the G12C inhibitors, but then eventually the tumor outgrows. However, when the same experiment is performed in mice with an intact immune system, a fraction of the mice can get cured. So this suggests that our own immune system, the anti-tumor immune response, the adaptive branch, both the innate and the adaptive branch of our immune system, contributes significantly to the efficacy of KRAS G12C inhibitors. So with this in mind, and we also know that when we treat mouse tumors with a G12C inhibitor, we inflame the microenvironment. Suddenly we see an influx of effector cells, of immune cells, as you can see here in C, which is being stained for CD8 T lymphocytes. And you can see that in response to AMG 510, we inflame the microenvironment. So these immune cells are going in and are playing a role in the clearance of the tumor. So with that in mind, it makes sense to try to combine immune checkpoint inhibitors that with KRAS G12C inhibitors, and you can see in B that by doing so, you are achieving synergistic efficacy and long-lasting cures, long-lasting responses in these mice, which are not achieved when you treat with each individual agent alone, Sotorasib or anti-PD-1 alone. But where you combine them, you can see that these tumors do not grow. So with that in mind, several trials for different KRAS G12C inhibitors have assessed the efficacy and safety of combinations of G12C inhibitors with inhibitors of both PD-1 and PD-L1. From previous experience with other targeted therapies, whenever a targeted therapy, especially drugs targeting the MAP kinase pathway, are combined with immune checkpoint inhibitors, one of the concerns is the safety. So as a way to overcome safety concerns and treatment-related adverse events, one strategy that has been employed in the past is to try not only different doses and different types of immune checkpoint inhibitors, but also different schedules of administration. In other words, concurrent administration, where you're giving the G12C inhibitor, the targeted therapy, together with the immunotherapy, or sequential or a leading kind of schedule, where you're starting with a G12C inhibitor and then you're incorporating the immune checkpoint inhibitor further down the line. So both of these approaches were tried in the multi-arm CodeBreak 101 clinical study, as well as arms from the CodeBreak 100 clinical study. The conclusion from these studies is, as you can see in C, the objective response rate across all different schedules of administration and dose levels was 29%. Encouragingly, the median duration of response was almost 18 months. So that's higher than what is observed with Sotorasib on its own. However, on the other hand, the drawback was that for several of these combinations, especially the concurrent schedules, when immune checkpoint inhibitor is given at the same time with a G12C inhibitor, we saw significant toxicity and in many cases prohibitive toxicity. And the main type of toxicity that is observed is hepatotoxicity, elevations in liver tests, which in many cases were grade 3 in this clinical trial. However, in the sequential cohorts and at lower doses of Sotorasib, the efficacy was improved. So right now, there is a cohort that's assessing sequential administration, so lead-in administration of Sotorasib at a dose of 240 milligrams with pembrolizumab, and with the pembrolizumab starting 21 days after the start of Sotorasib. That seemed to be a safer combination or schedule in this arm, and we'll know more. So this is going to be explored further. What about adagrasib in immunotherapy? So this has been tested in the first-line treatment setting in the KRYSTAL-7 clinical trial. And at least from the preliminary data that we have seen, the safety appears reasonable. It appears to be a tolerable combination, and preliminary anti-tumor activity also appears encouraging. So here, across all patients, the objective response rate was above 50%, was in the region of 55%, 11 out of 20 patients in this cohort. Obviously, the median follow-up, the time that we were followed patients in this trial was limited, so we need to see more long-term data, but this is certainly an area that, this is certainly something that will be looked at quite carefully. What other kind of approaches are being followed for incorporation of G12C inhibitors into the first-line treatment space? Well, There's two different approaches. So one is this standard approach with adagrasib on its own or in combination with pembrolizumab. However, the alternative approach is to use Sotorasib, not in combination with immune checkpoint inhibitor, but with platinum doublet chemotherapy, but limit the patient population to patients that are less likely to derive benefit from immunotherapy, that may require immunotherapy less. So these are patients in this study, for example, with lack of PD-L1 expression, PD-L1 0. And Amgen have announced that they are planning a phase 3 randomized control trial studying precisely this. So the combination of Sotorasib with platinum doublet chemotherapy versus the standard of care approach, chemoimmunotherapy, in previously untreated patients with a KRAS-G12C mutated non-small cell lung cancer and PD-L1 less than 1%. The other approach is to also target these co-mutational subgroups. For example, the subgroup with STK11 mutations that we know derives less benefit from the addition of immunotherapy to chemotherapy. And there are two clinical trials assessing this in the first-line space, the CodeBreak 201 clinical study, and the cohort within Crystal-1 is also assessing the efficacy of adagrasib monotherapy in patients with STK11 mutant non-small cell lung cancer. What about moving KRAS inhibitors to earlier disease stage? This is an area that I'm very interested in. So there are currently 3 clinical trials and perhaps more in development that are assessing different combinations of KRAS inhibitors in surgically resectable disease. The first one is a study that I'm leading here at MD Anderson. So this is a phase 2 study of neoadjuvant Sotorasib in combination with platinum doublet chemotherapy, either cisplatin or carboplatin and pemetrexed. This is for patients with stage 2A to 3B non-squamous non-small cell lung cancer with a KRAS-G12C mutation. And in this study, patients are going to be treated with Sotorasib and 4 cycles of chemotherapy. After that, they are going to go to surgery. And we're going to test 2 different dose levels of Sotorasib, but we're going to start with a full dose. And the second study assesses just Sotorasib, not Sotorasib with chemo, but Sotorasib alone. This is being led by Dr. Borkae at Fox Chase Cancer Center. This is in patients with stage 1B to 3A, single station N2 disease. And here Sotorasib is being given 960 milligrams once a day for 28 days prior to surgery. And the third study is being led by Dr. Maron, who I know is speaking later. And this is the NeoCan study. This is a phase 2 trial of neoadjuvant adagrasib with or without nivolumab in patients with resectable stage 1B to 3A KRAS-G12C mutated non-small cell lung cancer. In cohort A, patients get adagrasib at the full dose, 600 milligrams twice a day, just a single agent. And in cohort B, they get the lower dose of adagrasib, 400 milligrams twice a day, in combination with nivolumab for 6 weeks. So 3 doses of nivolumab given every 2 weeks, and then they proceed to surgery. So I'm going to spend the last few slides talking about emerging novel approaches. So I've told you about what is actively going on right now, but we're going to spend some time talking about novel approaches. And there are many, so I'm not going to do justice to all of them, but I'm going to highlight 3 different approaches that I think are interesting. So both Sotorasib, adagrasib, as well as the other G12C inhibitors that I mentioned up to now are off-state inhibitors. So in other words, they bind to the inactive form of KRAS G12C, which is KRAS G12C bound to GDP. However, there are conceptual advantages to targeting the on state of KRAS. We may be able to overcome certain mechanisms of adaptive resistance that are observed with off-state inhibitors if we were able to target the on state of KRAS. In other words, KRAS bound to GTP. So there are now several approaches to develop RAS on inhibitors, so inhibitors of the active state. And one that is advanced, that's kind of entering clinical development, is the tri-complex inhibitors. This is a strategy by Revolution Medicine. So this is based on novel chemistry. So these are inhibitors that bind to an abundant protein within the cell. It's a nucleophile, it's an immunophilin called cyclophilin A. And then the binary complex between the inhibitor and cyclophilin then binds to GTP bound RAS and creates a tri-complex, a ternary complex, if you want, that impairs the downstream signaling. So kind of shuts off signaling downstream of KRAS. And the advantage of this approach, and if you want the appeal of this approach, is that you can develop tri-complex inhibitors to different KRAS mutant alleles, not just KRASG12C or KRASG12D, but a variety of KRAS different alleles. And you can see that there are inhibitors in development for G12D, G12V, G13D, Q61, G12R, and G13C, and other. But also, there are inhibitors that are called RAS multi-inhibitors. So these are drugs that are able to simultaneously target multiple forms of mutant RAS, and not only KRAS, but also NRAS, other RAS isoforms, as well as wild type RAS, potentially affecting and reducing the ability, reducing adaptive responses that promote resistance to mutant selective inhibitors. And the phase 1/1b multi-center study of RMC-6236, this is the RAS multi-inhibitor in patients with advanced small tumors harboring specific mutations of KRAS is active and enrolling. One interesting approach is also the development of tools to not inhibit the KRAS, but degrade it and actually break it down, extinguish KRAS. And there are several approaches in development. Some of them are called PROTACs, proteolysis targeting chimeras. So these are drugs that are bifunctional drugs. So they bind on one hand to, if you want, a warhead such as adagrasib that is able to go and bind to KRAS G12C, but on the other hand, they bind to a protein within the cell, such as the VHL E3 ubiquitin ligase, and these are proteins that are able to target other proteins for degradation using the internal natural degrading protein degradation system that exists within the cells, and it's mediated by the 26S proteasome. So this is a very promising and emerging strategy. We will see, no doubt, we'll see much more of this in the future. And then finally, I'm going to do justice to data presented by my colleague, Dr. Negrao, yesterday in New York. So adoptive T-cell therapy approaches, and there are several different approaches, but I'm going to highlight the TCR T-cell therapy approach here because for the first time, the first patient that was treated with this mutation had an objective response. So this is a highly promising approach, a completely different approach using T-cell therapy to target KRAS mutant tumors. And no doubt we'll hear much more about this in the future. So I want to be mindful of your time. I'm going to mention the key conclusions and next steps. So what are the next steps? So we need to define optimal and tailored to the individual therapeutic combinations to enhance the efficacy of KRAS G12C inhibitors. We need to identify biomarkers of response to both monotherapy and distinct combinations of G12C inhibitors with other targeted therapies. We need to develop strategies to maximize the immune sensitizing potential of KRAS G12C inhibitors. What are the optimal immunotherapeutic approaches? We know that immune checkpoint inhibitors such as pembrolizumab is one approach, but can we target other components of the tumor immune microenvironment and synergize with KRAS G12C inhibitors? We need to see data on the first-line metastatic studies in combination with chemotherapy and/or immunotherapy, and we need to see data for KRAS inhibitor combinations in earlier disease settings. We need to understand mechanisms of de novo and acquired resistance, and of course, we now have novel mutant selective inhibitors, including inhibitors of the active KRAS state, as well as RAS multi-inhibitors, drugs that are able to target multiple RAS isoforms. And finally, we need to see new approaches to tackle KRAS using degraders to extinguish the protein rather than just inhibit it, as well as novel immunotherapy approaches, including both adoptive cell therapy as well as vaccines. I didn't elaborate on vaccines because I know that Dr. Marone will be talking about this in the future. So with that, I'm going to thank you and happy to take any questions. Sorry, I've taken a little bit more time.
Well, thank you so much for that amazing fire hose of information. I have probably about 1,000 questions that I could ask you, but I also need to stay on track on time. And so I'm gonna just ask you. Well, first off, I'm gonna mention to everybody that's watching that Dr. Negrao's information that was presented yesterday, we're going to have him come back to have a conversation with us in November. We've already got that scheduled. And if I could just get you to answer one question that's kind of like back a little ways. You mentioned that you do studies on immunocompromised mice and I wrote it, hang on a second, I wrote it down. Deficient and also competent mice. Do you often see a difference in the way they respond to different treatments?
Yes, absolutely. This is a very important question, and this is the whole base, the whole conceptual basis for combining KRAS inhibitors with immune modulatory drugs. So, in immune deficient mice, we see in response to a G12C inhibitor, you will see initial shrinkage in many models, but that shrinkage does not last. So eventually, the tumor finds a way to overcome the KRAS inhibitor and grows out. We only see long-lasting responses and potentially cures, what you would consider a cure in the mouse, in the context of an intact immune system. This suggests that our immune systems get turned on in response to a G12C inhibitor, and that the immune system becomes to a certain point responsible or contributing to the clearance of these residual KRAS mutant cells that may not be, that may not die immediately in response to the KRAS G12C inhibitor. So the efficacy of the G12C inhibitors is always higher in the context of an intact immune system. And this is why we want to combine G12C inhibitors or other KRAS inhibitors with drugs that either relieve immune checkpoints, such as the immune checkpoint inhibitors, or other drugs that modulate the immune microenvironment, for example, drugs that target immunosuppressive myeloid cells in the tumor microenvironment to further unleash the power of our own immune system against the residual KRAS G12C mutant cells that have not died as a result of the KRAS inhibitor on its own.
Okay, that leads me to the next question. Does it have an implication? I said one question, I lied. Leads me to the next question does it have anything to do with the co-mutations that we have? You mentioned ATM, STK11, KEAP1, those sorts of things. Are you seeing big differences and big swings, or is that something that's being concerned and being evaluated?
This is a fantastic question, Terry, and this is something that I'm working on in the lab quite intensely. It's actually one of the main streams in the lab. So yes, in answer to your question, the commutational status of the tumor impacts very much both the baseline immune profile, what it looks like before the G12C inhibitor, but also the immune modulatory effects of the KRAS G12C inhibitor. For example, we know that in STK11 mutant tumors, these are rich in myeloid suppressor cells. So these got few lymphocytes, which are the good cells that fight the tumor, and we've got a lot of myeloid cells that have got a suppressive effect. And it's actually KRAS to a large extent, KRAS signaling, that promotes this accumulation of suppressive cells. KRAS itself can be immunosuppressive. So when we treat these KRAS LKB1 or KRAS STK11 mutant tumors with a G12C inhibitor, we see a dramatic reduction in the number of suppressive myeloid cells, which no doubt contributes to the efficacy of these treatments. But this is a very important point because different combinations of immunotherapies or immune modulatory drugs might be more applicable to different commutational subgroups of KRAS G12C. So this is something that is no doubt will be important.
Okay, I think that's an excellent answer. And it seems like it just really pushes us as patients to have to learn how to advocate to make sure that we know not just like the one marker that we have, not just that we're KRAS G12C or D or whatever, that we have co-mutations and what those might be so that we can best identify things that will work best for us. So, okay, and I could talk to you all day, but we're gonna have you come back another time and for the community. So yeah, so that was my way of kind of like volunteering you that you'll be back. So we appreciate you doing that.
I would love to.
Okay, but at the same time, we're not going to let you slide away. Okay, we want to. We're engaging all our speakers to make sure that you kind of know our war cry, and that you do get a sense of who we are and what we do. And so we're kind of doing a count off with just the group that's here in the room, which is a little bit more limited as a factor of the hurricane. But we'd like to do another Kick Cancer's KRAS Live with you, engaging in along with the folks that are here. So if you're willing, I'm going to do like a count off, 1, 2, 3, and then we'll do a kick cancers KRAS all together. So if okay, everybody in 1, 2, 3, kick cancers KRAS. Thank you so much. We appreciate you.
Take care.